Here's the next part of the videos from Chapter 8. This is a video on meiosis. So if you remember, meiosis is a type of cell division to produce the sex cells or gametes, sperm and eggs, for sexual reproduction. So notice in sex there's an E, in meiosis there's also an E, okay? And in gametes there's also an E. That might help you remember the overall function of meiosis. So here we go.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with two sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of two identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of two sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into two cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
So a nice little visual there on meiosis, cell division to produce our gametes or sex cells. So one of the things to recognize in meiosis when comparing it or contrasting it with mitosis is meiosis 2, the second round of cell division in meiosis. Comparing that to mitosis, which only has one round of cell division, you should recognize that essentially the steps are the same. Okay, so take a look at mitosis and compare it to meiosis 2. And you'll see that what's occurring with the chromosomes is the same. All right, let's see if there's any more videos here in Chapter 8. Cute dogs. Oh, yeah, here we go. Genetic variation that occurs during meiosis through two processes. Random alignment of the chromosomes in metaphase 1, and then crossing over exchanging of comparable material from the homologous chromosomes from their chromatids, and that occurs in prophase 1. So let's watch this video.
In sexually reproducing organisms, 3 processes lead to most genetic variation: independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being, with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents.
One little comment here. When the homologous chromosomes, when they get close enough together and they match up so that they can exchange bits of genetic information on non-sister chromatids, basically swapping over as you see here, they're color-coded so you can see how they're exchanging bits of genetic information, and this occurs in prophase 1 of meiosis 1. Since there are 1, 2, 3, 4 sister chromatids involved in this process, this combination of 2 homologous chromosomes is referred to as a tetrad. That came up in lecture and or in your textbook because there's 1, 2, 3, 4 sister chromatids involved, and tetra means 4. So this is referred to as a tetrad. And there's another one down here. This association of homologous chromosomes is called a tetrad. Okay.
Chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
Okay, so the 3 sources of genetic variation in sexually reproducing organisms: crossing over, independent alignment, which is sometimes called independent assortment, and then the random fusion of gametes. Oh, see, here's that tetrad terminology. And crossing over.
During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of two non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes.
So this little video was a review of the one we had previously. So just a review. And that might be it for this chapter. Yes. Okay.